Thank you very much, Mantal. And our next speaker is going to be Professor J.J. Hu, who's going to talk about an inline photonic Raman sensor for accelerated pharmaceutical process development. J.J.
Okay, good morning, everyone. So here, I'm sharing with you a photonic sensing technology that can be used for inline process monitoring in the pharmaceutical industries. So the problem we are seeking to resolve here is the enormous amount of time and resources that's involved in today's pharmaceutical development process. Nowadays, on average, it takes 10 years and $3 billion to bring a new drug molecule to the market. And in this cycle, the process development step is critical because this is where the manufacturing process of the chemical molecules are being perfected. So if we have better chemical sensing technologies that allows us to better characterize the chemical reactions involved in the manufacturing, we'll be able to not only cut the cost, reduce the time, but also prevent any costly mistakes that can otherwise take place in subsequent manufacturing phase. So to understand what are the sensing needs from the community of pharmaceutical industry, we have conducted more than 170 customer interviews. And the messages we get from the potential customers are very consistent. The community is looking for sensing technology that can provide real-time information such that we can use it to monitor the kinetics of the chemical reaction. In addition, the technology should also be sensitive enough such that it can not only pick up the active pharmaceutical ingredient, but it can also detect any kind of potential impurity species. Last but not least, the sensor should be small and rugged. It should be small such that it's compatible with the flow chemistry configuration or small reactors. And it also must be rugged so that it can withstand harsh chemical, thermal, and high-pressure environments that's very common in pharmaceutical processing. So based on these requirements, we have developed our sensor technology, and what I'm showing this slide here is a CAD drawing of our sensor package. So we have liquid flowing through a sensor from one direction, and light propagate through another direction, and at the crossing point, this is where our sensor chip is located. So this configuration allows the light to directly interact with the molecules passing through in the flowing stream, such that we can provide real-time chemical information via the Raman scattering process. And here is the photo of the dissected sensor package that we have developed. And you can clearly see the photonic sensing chip sitting right in the middle of the package. And it's also connected to two fibers to enable light input and output on and off chip. Now, I want to emphasize that this actually represents the first demonstration of a packaged on-chip Raman sensing system. And this is significant because we have already experimentally validated that our sensor package is rugged and stable, such that it's fully compatible with the flow chemistry configuration that's now being increasingly adopted in the pharmaceutical manufacturing process. Now, I also want to mention that we have chosen to go with an on-chip sensing configuration rather than a conventional free space counterpart because of two reasons. One is that our photonic sensor chip can be readily manufactured using high-volume semiconductor microfabrication technologies, pretty much in the same way as the computer chips are being produced. Therefore, we can readily achieve scalable and high-volume manufacturing. In addition, our configuration allows the light to be guided in plane on the substrate for long distances. And therefore, we can significantly enhance the interaction between light and the molecules, and therefore boost the detection sensitivity. So we have thoroughly characterized the sensing performance of our sensor package here, and what we have experimentally demonstrated is that our sensor can provide more than 1 order of magnitude sensitivity improvement compared to state-of-the-art inline Raman sensors that's applied for the same purpose. So based on this information, we can then, here's how we envision our technology can be deployed in the pharmaceutical industry. So our sensing system will consist of one optical central control unit, as well as the sensing probes that are connected to the control unit via optical fibers. We believe that our technology can uniquely satisfy all the requirements from the pharmaceutical industry in that it combines real-time inline monitoring with very high sensitivity. It also features a small and a rugged sensor footprint, such that it's commensurate with the pharmaceutical manufacturing environment. On top of that, I also want to mention that our sensor also has the capability to perform multiplex sensing. So what that means is that we can use one optical central control unit to simultaneously monitor multiple sensing probes. So we can use it to detect different chemical reactions that takes place simultaneously in different reactors, or provide a spatial mapping of the ingredient concentration in a flow chemistry system to much better understand the chemical reactions. So to summarize, we believe that our high-performance inline micro-Raman sensor can be applied to significantly accelerating the development of life-saving pharmaceuticals. So if you're interested in our technology, I'll certainly be more than happy to talk with you at our poster, and I definitely welcome your input. Thank you.